Hello, everyone, and welcome to today's webinar titled Navigate Cell and Gene Therapy Success, Align cGMP Gene Editing with Regulatory Frameworks. This webinar is part of the Cell and Gene Therapy Virtual Event Series 2024, brought to you by LabRoots and is sponsored by GenScript. For more information on our sponsor, visit genscript.com. I am Israel Gonzalez of LabRoots, and I'll be your moderator for this webinar today. Before we begin, I would like to remind everyone that this event is interactive, and we encourage you to participate by both communicating with other attendees using the group chat and by submitting questions during the presentation. Now let's go ahead and get started. I'd like to introduce our speaker today, Daksha Patel, Senior Segment Marketing Manager, Cell Therapy at GenScript USA Incorporated. You can view Daksha's complete biography below. Now, Daksha, you may begin your presentation.
Thank you for the introduction. Hello everyone, I am Daksha from the GenScript. I am excited to share how the cGMP gene editing tools aligned with the regulatory compliance can accelerate success in cell and gene therapy field. Today I will cover the four key areas. One is first I will provide an overview of the cell and gene therapy and introduce the GenScript gene editing portfolio. Next, I will discuss the FDA regulatory requirement for CGT development, and then I will present some case study which showcasing how we providing the support to our client for gene editing solution, and that's aligned with the according to FDA guideline. And then finally, I will wrap up with the question and answer session. So let's start with the overview of the cell and gene therapy. Cell and gene therapy are advancing personalized medicine aiming to treat or cure the disease. While they have the same goal, they use the different method. Both the therapy use gene editing to change a patient DNA by adding or removing or altering the specific gene to treat genetic disorder. And this can be done into two ways. One is in vivo gene therapy, where new genes are directly delivered into the patient body, while ex vivo gene therapy, where is also known as a cell therapy and is involved in removing the cell, editing them in the lab, and then putting them back into the patient. And on basis of that, also cell therapy can be categorized into two type. One is the non-gene modified cell therapy, which use unmodified cell for treatment, and gene modified cell therapy, where the genetic material of cell is changed to enhance the therapeutic effect. And gene delivery can be done using the viral method, where the virus is used as a vector for delivering the gene, or non-viral method, which does not involve the viruses but use some other nanoparticle or the HDR template or some different kind of systems. And for this gene editing experiments, tool like CRISPR, zinc finger nuclease, and TALEN can be used. So let's focus on CRISPR-based gene editing. This groundbreaking technology has transformed gene and cell therapy by allowing the precise edit to DNA, making it possible to treat both genetic and non-genetic disease. Here shown is the key CRISPR system and their function. One is STEM if traditional CRISPR case. This is the most widely used system, like in CAR T cell therapy, which modify immune cell to fight the disease. And in gene therapy, it can also correct the inherited conditions such as the muscular dystrophy and hemophilia. Beyond this traditional method, it also used as the prime editing, in which is a more advanced method which can fix the specific gene mutation without cutting the DNA, and it's very useful. Like this is like sickle cell. The another way the CRISPR Cas technology can be used as a base editing, which perfect for correcting the single gene disorder like beta thalassemia by changing just one letter of the DNA. Uh, Another one is the R&A and editing technology, and this technique doesn't alter the DNA but adjusts the protein level, which is useful for treating condition like neurological disorder. And then last is the CRISPR activation or CRISPR interference. And this method can either boost or silence gene activity temporarily, which is great for immune therapy or regulating the gene involved in the tumor suppression. So CRISPR is now heart of both in vivo and ex vivo therapy and driving innovation and offering the new hope for treating the range of disease. Let's now explore into essential gene editing tools that power these techniques. As a leading provider of nucleic acid for CRISPR, GenScript offered the broad range of CRISPR solution to support the various gene editing and therapeutic need. Our gene editing portfolio is built to meet the expanding demand of cell and gene therapy. And here are represented some important reagent and service offered by GenScript, which include the sgRNA design tool as well as HDR design tool, CRISPR library, CRISPR plasmid, CRISPR cell line, knock-in optimization kit. Even we providing different kind of nucleus, Cas9, Cas12, Cas13, Cas9 mRNA, and different type of sgRNA and different type of donor DNA template. While we have the wide range of products and services, today I will focus on two main areas. First, we will discuss the CRISPR component used for knockout application, specifically the single guide RNA and Cas9 protein. And second, I will cover our DNA knock-in template, which include the various HDR template designed for different application. Let's begin with the CRISPR RNP for gene knockout. So the process is straightforward. Chemically synthesized guide RNA is combined with the Cas protein to form the RNP complex, which is then delivered into cell to knock out the gene. And using chemically modified single guide RNA offers several advantages. One of the key benefit is a faster manufacturing, as synthetic sgRNA can be produced in just 3 business days, unlike plasmid or viral method which take week or more. And since there is no foreign DNA is delivered into cell, there is no risk of unwanted integration into the cell genome. As well as editing happening faster because synthetic sgRNA doesn't need to be transcribed inside the cell. The Cas sgRNA RNP is only present temporarily and breaking down quickly after editing, which reduces the risk of off-target effect also. And this method also work well in difficult to transfect cell and achieving the high editing efficiency. Additionally, synthetic sgRNA can carry sequence up to 200 nucleotide long, offering high packaging capacity. And another advantage is its low immunogenicity, as synthetic sgRNA trigger fewer immune response and less toxicity compared to sgRNA made from plasmid or viral method, making it ideal for in vivo study also. So, RNP based CRISPR knock in is a gene editing technique where RNP complex target a specific location on the genome, creating the double standard break that allow for the insertion or of desired DNA sequence through homologous recombination or non-homologous end joining. In this approach, the RNP complex and HDR knock-in templates are designed, manufactured, and introduced into the cell simultaneously using the electroporation. Afterward, the cells are analyzed for downstream application. And this technique is widely used for precise genome modification and research and therapeutic application, particularly in cell therapy and gene edited model development. A key advantage of using HDR template include the functioning as a non-viral delivery system, reduce the off-target effect, efficient delivery, and quick response. By utilizing this modality, one can significantly improve knock-in efficiency while also minimizing the cost making this approach more accessible for various applications. Uh, Success in CRISPR-based gene knockout or knock-in rely on the Cas9 protein quality, which act like the molecular scissor to cut the DNA precisely. As CRISPR moved from basic research to clinical manufacturing, the need for high-quality material and comprehensive documentation become crucial. To address to address this need, GenScript offered the validated GMP and REO grade Cas9 protein, Cas12, and Cas13 nuclease. These products are manufactured under strict GMP guideline, ensuring the aseptic production, complete traceability, and thorough testing. In addition to high quality material, GenScript provide full documentation, facilitate the site audits, offer the extra support to help them meet regulatory requirement in the different region. Smartly designed synthetic sgRNA are proven effective for targeted gene editing in knockout and HDR-mediated knock-in experiments. To support the RNP-based CRISPR knockout and knock-in, GenScript provide two type of sgRNA. One is the easy edit sgRNA and another one is the safe edit sgRNA. Easy edit is more economic solution, shows the high editing efficiency and flexible at the scale. While safe edit is sgRNA is HPLC purified, reduce the off targeting and show minimum impact on the cell viability. We also provide the free online bioinformatic design tool to designing the sgRNA and HDR template to accelerate the precise gene editing. Additionally, GenScript also provide add-on QC options like microplasma testing, endotoxin testing, stability testing, cytotoxic testing, and as per the demand. To support the RNP-based CRISPR knock-in, GenScript provide the high-quality sequence-verified HDR template both in linear and circular format to enhance the efficiency of CRISPR experiments. And we can support which can support the various non-viral application. One of them is the gene exact single standard DNA. It has the low toxicity and ensuring the high efficiency and minimizing the off-target effect and making it perfect for T cell therapy and generating cell or animal model. And gene exact can insert the length of up to 5 kb, making it versatile for many applications. Another one is the gene wend dsDNA, which is a double stranded DNA with close end structure. And this covalently close end and the linear DNA, mm, linear DNA enhance the DNA stability and reduce the risk of non-homologous end joining and thereby improving the accuracy of knock-in. It is ideal solution for large-scale screening, process optimization, and scale-up in cell and gene therapy, and it can be allow the inserting up to 10 kb. Another one is the gene circle, which is new plasmid like small circular double stranded DNA vector with very small backbone of approximately 430 base pair, and it has no antibiotic resistance gene, and it serves as an efficient knock-in template and shows the low toxicity and is a great alternative for traditional plasmid HDR template. It can be utilized in various applications such as the knock-in template, viral packaging plasmid, or non-viral vector. And it can insert the length up to 20 kb. Overall, this option provide efficient solution for CRISPR technology for effective gene editing with minimum side effect. And our products are specifically designed to minimize the toxicity, and it's also allow the scaling up, supporting the future CMC work and making them highly suitable for GMP manufacturing. Importantly, we have the manufacturing capability for all the three HDR modality, sgRNA, mRNA, from discovery phase to the GMP grade production. To meet the growing demand of reagent for both research and clinical grade, we expand our manufacturing facility in 2023 to over 4,000 square feet. And our facility is specifically designed to minimize toxicity and facilitate the scaling up, creating the opportunity for future CMC work, as well as making our product highly suitable for GMP manufacturing. Our grade C manufacturing room is equipped with grade A isolator for sterile filtration and aseptic finishing to support the sterile production. Alongside our hardware and workflow, we have fully integrated quality management system that can ensure the compliance throughout the manufacturing process and help with the IND filing. Our comprehensive QA and QC documentation system guarantee that all requirements are met as per the guideline. And in this plant, we have the 8 single guide RNA manufacturing line capable of supporting milligram to gram level GMP manufacturing. We also have 4 manufacturing line for linear single standard and double standard DNA, accommodating up to 100 milligram of GMP scale. And additionally, we have 2 manufacturing line for gene circle with capability to gram level GMP manufacturing. And our facility is designed to make our product highly suitable for both research grade as well as GMP manufacturing. And key difference between the RUO and cGMP lies is the facility design and production capability. For RUO manufacturing, we utilize the ballroom concept where all the instrument for synthesis and purification are placed in one room, and this allow flexibility and efficiency in the research phase. And in contrast, for cGMP manufacturing required each step occur in segregated room to maintain the strict regulatory standard. For instance, if purification is conducted in a clean room, it take place in designated area, while for GMP product requires sterile condition. Therefore, the grade A isolator in the GMP environment is specifically designed for the aseptic finishing process. When it comes to the capacity, the average lead time and scale differ significantly between RUO and GMP. And in the workshop, our only one sequence is manufactured at a time. In summary, our facility is well equipped to minimize the toxicity and facilitating the scaling up and making our product suitable for both RUO and GMP stage. Uh, as of last month, like we have delivered over 120 clinical batches and completed more than 30 global audits. And we take pride in supporting our 50 IND projects, 9 of which have received global IND approval. To achieve these results, we ensure the smooth transition from research to um, GMP products, which help speed up our process. The REO phase is the primary design for discovery purpose, focusing on screening gene of interest, conducting the individual functional assay, and the small scale validation. During this phase, we implement an ISO 9000 quality management system. And as we move into clinical development, such as the animal study or toxicological study, we transition into GMP like or cGMP quality management system. And for IND filing as well as for during clinical trial and commercialization, we adopt the full cGMP quality management system. To support our client, our GMP quality management system is designed to compile with ICH and FDA regulation. This system includes the 6 subsystem: quality assurance, quality control, production, facility and equipment, material and personnel. We implement the appropriate quality management system with some flexibility allowed during the early phases. However, when it comes to IND filing and critical phase, we require more validation work and increase the process control. For example, both the analytical method and the manufacturing process must be validated before the commercialization. And with this quality management system in place, we can significantly reduce quality cost, making our manufacturing process more cost effective. Additionally, GenScript offers extensive support at various stages of development. During the preclinical phase, our scientists work closely with client on gene editing design. We utilize online tool for sequence design and engaging in technical discussion. As the project progress to IND filing, we assist in preparing submission document into the FDA or and also attend the pre-IND meeting with the FDA. And furthermore, we maintain the drug master file for single guide RNA, which facilitate the compliance and help to address FDA comments during the clinical trial. As CRISPR technology developed, it's important to understand the regulatory requirement. And in this section, we will discuss the FDA official guideline for CRISPR therapy and the related regulatory requirement. Following this guideline is crucial to ensure the CRISPR therapy are safe and effective. This visual shows that regulatory requirements set by the FDA to ensure the safety and effectiveness of the gene edited product. The FDA guidance document like human gene therapy product incorporating human genome editing uh, and recommendation for CAR T therapy outline the entire product life cycle from design to clinical trial. And this guideline highlight the need of align CRISPR technology with high quality material which meets the IND and GMP standard. And the key FDA recommendation for gene editing include defining the type of genetic modification needed for the desired therapeutic effect, identify the method to achieve the required DNA sequence change, selecting the appropriate delivery method for gene editing component, and then using high quality GMP grade material into the creation of genome edited product. The FDA emphasized in the importance and sourcing and managing gene editing component, and developers should always use the best reagent at every manufacturing stage. While it is okay to use phase appropriate gene editing reagent during the different development phase, the quality and manufacturing process of this component must be confirmed before major clinical study. And its significant change occurs, managing the associated risk should be high priority. And comparability study must be supported by strong scientific reasoning. It is crucial to ensure that the key characteristic of gene editing reagent, um, like purity and editing efficiency, remain consistent before and after any change. In summary, as per FDA guideline, gene editing component, including the editor, guide RNA, donor DNA template, must be of the highest quality throughout the product development. While phase appropriate reagent can be used at different stage and any significant change required, careful risk management and comparability study to maintain the product integrity is a must. So depending on the application, genome editing component like sgRNA are categorized into three different scenarios. When one is in vivo application, when sgRNA is encapsulated with mRNA and injected directly into patient, the sgRNA classified as a drug substance. For ex vivo application, example universal CAR T cell therapy, where cell isolated, treated with CRISPR, and then reintroduced. In this case, sgRNA is considered as a critical component. And like iPSC stem cell based therapy, in this context, the sgRNA is treated as a starting material. In all this scenario, the FDA mandates that the sponsor focus on the several key aspects. First is the stability, identity, purity, and activity of gene editing component must be thoroughly evaluated. Second, the monitoring for processed residue or impurity is crucial. Third is the analytical method used to assess the gene editing component must be qualified and appropriate process control should be implemented during the gene editing component manufacturing. Additionally, long-term stability study are critical, particularly in support of clinical trial and eventually commercialization release. Finally, a phase-appropriate GMP quality management system must be in place with with facilities designed to prevent any cross-contamination. Let's take single guide RNA as an example to better understand this. The FDA updated requirement now specify that purity level of full length product must be at least 80%. It is also crucial to identify and control any impurity that may be present. In terms of purity, and any impurity greater than 0.1% must be identified. Additionally, for sequence confirmation, NGS is recommended as the identification method to ensure the corrected full length of sgRNA sequence is present. There are also an emerging requirement to use NGS as an orthogonal purity assay to identify and quantify the sequence variant. And this dual approach ensures both sequence confirmation and purity testing, which are crucial for addressing the safety concern. In addition, residual solvent and elemental impurity introduced during the manufacturing process must be assessed due to the safety concern. Moreover, stability, particularly at the recommended stage of temperature, need to be evaluated to understand the long-term effect. So FDA guidelines also highlight the importance of having long-term stability data for single guide RNA. Let's now understand some real case study and how GenScript support the different customer at different stage. So during the chemical synthesis of single guide RNA, some side reaction are unavoidable. For example, of one such reaction can cause G2A mutation. If G2A mutation occur, it disrupt or interfere with the base pairing and increase the potential for off-target effect. However, this mutation may seem minor and even undetectable when using the mass spectrometry because the difference in molecular weight is only about 1 Dalton. And traditional analytical method like UPLC, HPLC also struggle to distinguish the G2A mutation because the change is too small. In contrast, the NGS sequence offer the superior detection capability, allowing us to identify the G2A mutation and allow us to make this as our quality control improvement. After preparing like NGS library, we confirm the reproducibility test for G2A mutation as shown in the sample testing. And once we confirm that NGS is a powerful enough, we conduct more thorough study and we intentionally overkill the side reaction to confirm the G2A mutation. The orange line represent the overkill reaction where all the side reaction have been amplified and clearly showing that G has changed to A. After optimizing the manufacturing process, we see that all the green line indicate that G2A mutation has been completely eliminated. And this demonstrates that NGS is more powerful analytical method than mass spectrometry in identifying G2A mutation and difference and guiding us to upgrade our manufacturing process. And the second part of the case study used NGS for orthogonal purity testing. Building on our previous experiment with NGS, we further improved the analytical workflow by adding a unique molecular identifier to the library preparation process. Using this technology, we can differentiate more accurately between the n-1, n+1 endpoint mutation during the analysis. Here is an example how we conduct the NGS-based purity testing. In this case, the full-length product analyzed by NGS show about 80% purity, 85% purity, and all the potential sequence variant are listed here with as low as 0.5%. The FDA required impurity greater than 1% must be reported. And as you can see, nearly all these variants can be identified using NGS method. Here, uh, we use the NGS orthogonal purity testing for impurity profiling and purity testing. This is the real case study. We work with one of our client. We black out the guided region and found that using NGS, we could detect the mutation, including the base mutation and N+1 mutation at different level. Using in silico prediction, we assess whether this mutation would have any off-target potential in gene editing experiments. And we discovered that certain mutations such as A2U and U2A have the higher potential to cause off-target effect. To confirm this, we perform the weight lab test. We synthesize all the potential mutation along with the full length product and positive control to conduct on target efficiency test. From this test, we found that the two mutations significantly reduce the on target efficiency and increasing the potential for off target effect, and we identify them as a critical impurity, which is highlighted in the with the yellow line. And this impurity is U17A as well as A02U. And to further identify the maximum tolerable residue level of each impurity in the final product without increasing off-target editing, we spike impurity, both the impurity which have the highest risk of off-target to the full-length product at the different ratio. And we use this sample to perform CRISPR gene editing in primary T cell and assess the editing efficiency of each sample. Uh, when we spike at ratio of 50%, the A2U mutation resulted is less than 4% of target effect as shown in the figure A, while the U2A mutation at the same ratio cause almost 70% of target effect as shown in the figure B. And this demonstrates the power of NGS as a purity testing method, assisting us to identify critical impurity and guide us to improvement in the manufacturing process. Another case study is focused on detecting the residual sgRNA into the cell. The US Pharmacopoeia mandate the detection of residual sgRNA. In addition to traditional method like HPLC and mass spectrometry, we developed the qPCR based method specifically for identifying the residual sgRNA even within the cell system. And the use of qPCR facilitate the monitoring of sgRNA residue, ensuring uh, safety and efficacy in the cell and gene therapy application, which is critical for compliance with the regulatory standard. qPCR method utilizes the Tuckman probe, which has the fluorescent tag on the one end and quencher on the other hand. And when the probe binds to the sgRNA sequence, the fluorophore is cleaved from the quencher and generating the fluorescent signal that allow us to measure the residual sgRNA level. We applied this analytical method to the T cell system, introduced only sgRNA, which is represented as a black line, and the uh, RNP complex, which consists of the sgRNA and Cas9 protein, represented as a blue line. In both the group, There was a significant decrease in residual sgRNA level, reaching the plateau after the 48 hours. And this shows that Cas protein can stabilize sgRNA more effectively, leaving more residual sgRNA compared to the pure uh, sgRNA into the cell system. And this analytical method allows us to determine residual sgRNA level, which can be crucial for safety evaluation in the clinical trial. And the pharmacopoeia required the extraction accuracy within the range of 50 to 150%, and relative standard deviation is less than 30%. In our test, we comfortably meet both the criteria, and we can detect the sgRNA at level as low as 2.5 picogram. The next case study is about the risk assessment. This case demonstrates how we perform the risk assessment together with our client, showing that HPLC traced data for sgRNA, where some impurities were categorized as early and late. We collected eluent during the purification process to investigate which eluent must be more harmful to the cell or affect the editing condition. This eluent were sent to our client and who performed the risk management assessment test on different sample and including the mock group, early eluent, main peak, and late eluents. And they evaluated the cell viability and editing efficiency. Based on that data, we confirmed that the early eluent was less favorable in the purification process. As a result, we adjust our purification protocol, particularly mobile phase and the gradient, to reduce or eliminate the early eluent. And this is a case study where we adopt the cost-effective control strategy during the early phase for iPSC-based manufacturing process for CAR iPSC allogenic therapy. Since iPSC manufacturing is a long process, from cell banking to the drug production, single-guide RNA used alongside the protein and donor DNA is considered as a critical raw material. And to manage this, we adopted GMP like quality management system, and this include the segmented clean room environment, environmental monitoring program, sterility control, cleaning and standardization of SOP, precise equipment and analytical method. And in collaboration with the customer, we designed this cost-effective manufacturing workflow for iPSC-based application. During the process, we made sure to address the quality issue and optimize the resources used and maintain the control. And this highlights that we work together with our client to meet the regulatory requirement and maintain the cost efficiency. In summary, to meet the regulatory requirement, GenScript support customer every stage of development process. We have different kind of quality control process is already in place, and we keep changing them as per the requirement of the customer. And during the preclinical phase, our scientists work closely with our customer uh, team on gene editing design. We provide online tools for sequence design and engaging in technical discussion to optimize their project. As customer prepare for IND filing, our scientists can participate in pre-IND meeting with the FDA and help to compile the IND submission document. For single guide RNA, we have submitted the drug master file that our customer can refer. And once the IND is filed, the critical trial design, GenScript as the manufacturer, focus on optimizing critical quality attributes and consider the scaling up for the future cost saving. If there are significant changes to the chemistry, manufacturing, and control process, GenScript submit the necessary revision to the FDA. As the customer approach into the biological license application phase, we assist them with the BLA documentation and support them in responding to the any FDA comments. Overall, GenScript ensure consistent manufacturing, and collaborates with the customer on major changes to assess their potential impact on safety and efficacy throughout the different trial phases. So, GenScript Life Science Group offer custom tailored workflow for key market segment like gene therapy and cell therapy, from early stage discovery, lead identification, optimization to preclinical and clinical stage. And we provide the right solution to the support your journey, jour- your jour- journey and towards the success. Thank you all for attending this webinar. And now let's move on to the QA session and address any question you may have.
Thank you very much, Daksha, for that informative presentation. Let's go ahead and dive into the Q&A segment of the webinar now. And we encourage you to continue submitting your questions for our speaker during this time. We have some questions coming in already. Looks like our first question is this. What advantages does NGS sequencing offer over mass spectrometry or HPLC when it comes to impurity profiling?
Great question. Like impurity profiling is critical because it helps identify all the potential side products and impurity along the target products such as N-1 or N+1, and sequence substitution in the single guide RNA may affect number of factor like so that's why next generation sequencing offer the several advantage over the mass spectrometry and HPLC. For example, it has the greater sensitivity. NGS can detect even small or indirect changes in the genetic sequence, such as the point mutation, and that may be not noticeable with MS or HPLC. Another one is the like it can do precise in mutation detection. So NGS can identify specific genetic mutations such as the off-target effect in gene editing, uh, um, which MS and HPLC maybe might miss. And as those methods are less sensitive to the small genetic change like G2A mutation, NGS can catch up on that. The NGS allow the high throughput analysis because it can do simultaneously sequencing of million of molecule and providing the comprehensive overview of impurity at the nucleotide level. And it's not required like prior knowledge, because in MS or HPLC, which often require the prior knowledge of the impurity for identification, and NGS can uncover unexpected or unknown impurity through its open-ended sequence-based approach. And last but not least, it's like NGS provide qualitative and quantitative information uh, along with the purity and impurity profile. So in summary, I can say NGS offer more detailed, sensitive, and comprehensive impurity profiling as well as purity profiling, which highly impact affecting in gene editing. So that's way people can design more uh, effective sgRNA sequence and use during their cell therapy or gene therapy product development.
Thank you for that wonderful answer. Our next question is, in what ways do residual sgRNA and impurities impact gene editing efficiency and off-target effects?
Yeah, as we covered in discussion, like residual uh, um, impurity can significantly impact both the efficiency and specificity of genome editing. Uh, Example, like in editing efficiency, if there is a impurity is present, they interfere with the formation of Cas9 sgRNA complex and leading to the lower rates of successful gene editing. And impurity can also increase the off-target effect. It may cause non-specific binding to the uh, complex of genomic sites and leading to unwanted mutation. And it's also maybe cause like activation of off-target cas activity, so potentially damaging the healthy cell or introducing the intended mutation into the at the end application. So typically, the product contain overall 80% um, purity, which is greatly recommended by FDA. So according to the um, overall guideline, it's very necessary to minimize the impurity for reducing the off-target effect as well as increasing the on-target efficiency and reduce the cytotoxicity and side effect of the cell or gene therapy.
Awesome. Thank you so much for that. And it looks like we have time for just one more question. How does GenScript ensure protection against cross-contamination during the manufacturing process?
Oh, that's good. So, like GenScript set up or develop some strategy to protect against the cross contamination to ensure the integrity of the products. So, first policy which is in place is the single use policy. So, GenScript has the policy in place where all the consumable, which is use and throw type, and even the raw material also using in the batches, so it will not get cross contamination. And then strict quality control is also in place. So how it's helping, that include the use of high purity reagents and consumable, and this ensure that all material used in this process meet the high uh, standard for the from the beginning of the reaction. And the another one is like GMP. GenScript follow the good manufacturing regulation, uh, which include like clean room and control environment, and that's also ensure that manufacturing process are consistently producing the high quality product. And we implementing the automation system also, so that's why it's minimize the human intervention, which further reduce the risk of contamination. And these measures collectively help GenScript to maintain the high safety and quality standard in the end product. But if you have any, if you're looking for more information, feel free to contact us, send us email. We will definitely love to connect with you and give you more detailed idea about how the GMP facility is managed and what are the different kind of regulation we are applying to reduce the cost contamination and maintain the best product production.
Understood. Thank you so much for that. And Daksha, thank you so much for your time and for your important research for this presentation today. Any questions that we didn't have time for today, as well as those submitted during the on-demand period, will be addressed by our speaker via the contact information that you registered with. And this webcast can be viewed on demand for 1 year until October 16th, 2025. And LabRoots will alert you via email when it's available for replay. We encourage you to share that email when you receive it to your colleagues and friends who may have missed today's live event. Thanks again to our audience for joining us today and to GenScript for sponsoring this webinar. Until next time, goodbye, everyone.
Thank you, everyone. Bye.